In this video, we're going to talk about Lab 6, which covers microscopy and mitosis. And so what you're going to do for this lab is you're going to use the following slides and instructions in your lab manual to complete your lab. Make sure that for each of the drawings for the mitosis part and also for the microscopy slides, please draw only one cell in each box and fill about 3/4 of each box with each drawing, meaning you want your image that you're drawing to take up most of that box. Also, a summary of what to draw and label is provided for you on page 85. So make sure that you refer to that page and that you cross off that you've labeled the following parts. So in the lab, we would be using what we call a compound light microscope. And what that means is that this microscope has two sets of lenses. It has a set of lenses up here, and these set of lenses are referred to as the ocular lens. Ocular referring to the eyepiece. So in the eyepiece, we have a set of lenses here. On our microscopes, these ocular lenses magnify an image 10 times what we see with our naked eye. We have another set of lenses down here, and this other set of lenses is what we call the objective lens. And on the microscopes, you're going to see objective lenses that are varying sizes. The smallest lens being the lens that magnifies the least. In most microscopes, that would be a 4x lens. We have a 10x lens, we have a 40x lens, and we'll talk about total magnification in a little bit. And so how we would use the microscope is you'll notice that on this microscope, there's this black platform. And this black platform is what we call the stage. On the stage, you can see these silver clips here. Those are called stage clips. And those are what are going to hold the slide on the stage. And so what we would do is we would take our slide and we would slide it and put it on the stage and make it fit within the stage clips. Now, without looking at our microscope, we would use these stage adjustment knobs to center our slide over where the light is. Now, I need to back up and tell you about how to turn on the light. So, couple things that you would want to know. On the microscopes, there's an on-off switch. There's another little knob here, and that's called the light intensity knob. It's also referred to as the rheostat. And this controls the amount of light that comes up from the lamp. So before we even begin to use our microscope, before we even plug it in, we would want to make sure that the on-off switch is in the off position. We would also make sure that the light intensity knob is as low as it possibly goes. The reason for that is we don't want to plug in our microscope and have the lamp already be on and send a big surge of power to the microscope, which could blow the lamp. So we want to make sure before we even plug it in that the on-off switch is in the off position, the light intensity knob is all the way down, so the lowest it can go. Then we would plug in our microscope, okay? And so behind here is where we would have our plug. We'd plug in our microscope. We'd turn our on-off switch on. We would then adjust the light intensity to about a 4. And what you would see when you do this is that you should be able to see some light coming up through the lamp. So the light's going to come up and it's going to pass through the slide that's on the stage. So it's important that when you put your slide on your stage, that you need to center your specimen over where the light is, right? If the light is hitting a part of the slide where the specimen is not, you're not going to be ever looking at that specimen. So it's important that you center your specimen over where the light is coming up. Now, when you start, you want to always start with the lowest magnification in place. So that means that on this, we call this a revolving nose piece. This piece here rotates, this black rubber piece. And this revolving nose piece, when it moves, it's going to switch different objective lenses into place. When we start, we always start with the smallest lens, which is going to be our 4x objective. And so we would put the 4x objective over where the light is. And so that's how we would always start. The 4x lens you're going to see in a minute is called the scanning lens. It's the one that we start with to try and find our image. Now again, the nose piece is this rotating disc that holds and switches the objective lens. Over here, you see two sets of knobs. The bigger knob that's closer to the base is referred to as the coarse objective or coarse focus knob. This smaller knob here is referred to as the fine focus. Now, the difference between these two is that both of these knobs are there to move the stage up and down. So if we use the coarse focus, it's going to move the stage much faster. If we use the fine focus, you can think of that as fine tuning. It's going to make the stage move smaller amounts, meaning it's not going to move the stage as much. So what we would do is we'd start with our coarse focus, and without even looking at our microscope, as making sure your 4x objective is in place. So without looking in your microscope, you would use your coarse focus and you would move the stage all the way to the top. Now, the reason we usually start at the top and not at the bottom is that our image is usually close to the top. You could, in theory, do this with the stage starting low and then work your way up, but it's going to take you a lot longer to find your image. So you want to start with the stage up to begin. You would want to adjust the width of these eyepieces. So these ocular lenses move, and you would want to make it so that you could focus, you could put your eyes into both eyepieces and only see one image. If you're looking in your microscope and you can't focus with both eyes simultaneously, that means that the width of the ocular lenses is not adjusted properly for your eyes. Everybody has different spacing between your eyes, and so you need to adjust the width of the ocular lenses so that you can comfortably look in those ocular lenses and see the image with both eyes simultaneously. So you can adjust the width of the eyepieces. Now, when you're looking in your ocular lens, right, your slide is on here, you look in your ocular lens, and you would start by using the coarse focus, and you would begin to lower the stage until you start to see your image come into focus. Once you can start to see your image, stop. You don't want to keep using the fine focus. So once you start to see your image, stop with the coarse focus, and then you would use the fine focus. The fine focus is going to make smaller movements to the stage. And again, it's going to allow you to fine tune your image. And so use your fine focus, and you would do that to get it into focus so that you can see it clearly. So we need to talk about our total magnification. And so we're going to write on here. So I said that our lenses, our ocular, is always 10x. So that's going to be constant. Our objective magnification, so our smallest one is 4x, and we call that lens the scanning lens. And that's because when you're at the lowest magnification, you are the most, think of it like zoomed out or zoomed in. Like when you use your phone and you pinch your screen and you can either zoom in or out. When you magnify the lowest, you're going to see the greatest field of view, meaning you're going to see the biggest area. So if we're using our 4x and our ocular is 10, to calculate our total magnification, we take objective times ocular. That gives us our total. So our total magnification is 40x. The next lens that we have is our 10x, and we call this the low power. So this is our low power lens. And so our total magnification for this would be 100x. The next objective is our 40x. And our 40x objective is called our high dry lens. The reason that it's called the high dry, so it's a high power lens, but it's dry. And what that means is that it doesn't use oil. Some microscopes will have a fourth lens on it, and that fourth lens will have an objective magnification of 100x. That lens, that 100x objective, would be referred to as the oil immersion lens. You have to use immersion oil in order for that lens to work properly. So if we're using our high dry, 40 times 10, our total magnification is going to be 400x. So when we do this in the lab, what we would normally do is we would use a letter E slide to get you to be familiar with your microscope and how it works. And so what that would look like is, this is my slide. There's a part you can label. And on the slide would be a small letter E. So on your slide, when you put it on your stage, you would do that so that your letter E is facing the right way. So that when you're looking at it on the stage without looking in the microscope, it looks the proper orientation of the letter E. However, if you were to look in the microscope, so here's your field of view, and this is your 40x. So we're looking with our scanning lens, our lowest possible magnification. What you would notice about that is your letter E would look like this. So it's not only flipped horizontally, meaning it's not only flipped from left to right, but it's also flipped vertically as well. So it's flipped in both orientations. It's both upside down and backwards. The reason that the letter E looks like that in the microscope is because we have our compound microscope. We have two sets of lenses. We have the first set of lenses, which is going to be the ocular lens, the one that's by the eyes. Then we have our objective lens, which is on the revolving nose piece. We have the specimen, which would be on the slide. And then beneath that, we have something called the condenser, which we'll talk about in a minute. So one of the things to keep in mind whenever you're looking at a microscope, whatever you see in the microscope, you have to think that in real life, what is actually on the slide is inverted to what you see when you look in the microscope. So that's just something to keep in mind when you're looking in the microscope, that in actuality, what is really on the slide is inverted because our compound light microscope has two sets of lenses. So now, we've focused on our slide using our lowest magnification. So our total magnification would be 40x. We're using our 4x objective, but we might want to go up a magnification, meaning we want to magnify the image more so it makes it easier to see. So when we go up a magnification, there's a few things that we need to do. And the first, before we even change the magnification, the first thing you always want to do is that you want to go ahead. Let's put my laser pointer back on. So what you would want to do is you'd want to use that stage adjustment knob. One of these moves side to side, one of these moves front to back. And you would want to make sure that when you look in your ocular lens, that your specimen, if you were using, let's say, your letter E, you would want to make sure that your specimen is centered. So before you go up in magnification, you need to recenter what you're looking at. If you don't, if it's off to the side and you go up in magnification and now you're only seeing a portion of it, well, now you've lost your specimen. So before you even change your magnification, you would need to first use your stage adjustment knobs, recenter your specimen. Make sure whatever you're looking at is in the middle of your field of view. After it's centered, then you use the revolving nose piece and you would rotate the 10x objective into place. When you do this, you only use the fine focus to fine-tune your image. You would not use your coarse focus. You would only use your fine focus. And you're going to see why in just a minute. But one of the things that you want to know, one of the reasons we should only need to use the fine focus is that our microscopes are what we call parfocal. And that means that the microscope stays approximately in focus when switching between objective lenses. So our microscopes are good quality. They should stay roughly in focus as we go from one objective lens to the other. Now, one of the things that you would know when doing this is that you would notice when you go up in magnification that your light intensity starts to decrease. And again, I'll draw why in just a minute. But there are several ways to adjust the amount of light. The first and the easiest is to use this light intensity knob. And again, that light intensity knob is called the rheostat. You can think of it like a dimmer switch. It's going to control the amount of light that comes out of the lamp. So it's your dimmer switch. Now, also, right beneath the stage, you'll see this black ring. And that black ring is what we call the condenser. And the job of the condenser is that it's going to gather and focus the light to illuminate the specimen. So it's going to take that light, which is really diffuse, and it's going to bring it to a point. So it's going to cause that light to go to a point. And so it's going to gather and focus the light to illuminate the specimen. There's a knob on the other side of this that you usually don't have to move often. On this black box on the other side, there's a little knob, and you can adjust the height of the condenser. And so normally we would start with the condenser as high as it would go. Usually we don't adjust the condenser to adjust the light intensity. That's probably the least common way to adjust the light intensity. So typically we start with the rheostat at a level 4, which I've already talked about, and we would also open the iris diaphragm. Now, what is the iris diaphragm? Well, on this condenser, there's a little lever, and that little lever controls the amount of light that exits the condenser. So the condenser's job is to condense the light. The iris diaphragm is like closing or opening the lens. So it's basically, if we close the iris diaphragm, it's going to let less light out of the condenser. If we open the iris diaphragm, it's going to allow more light through. And so if we go up in magnification and it begins to get darker, then that means that we're going to use the iris diaphragm. We're going to open it to allow more light in. Now you might wonder, well, why don't we always keep the iris diaphragm open? And the answer is that some specimens that are really faint, for example, if we were to look at blood smears, oftentimes blood smears are very faint and they're difficult to see if the light intensity is all the way up. If we close the iris diaphragm a little bit, it gives us a little extra contrast because not as much light is coming through. And that extra contrast allows us to see those faint specimens. And so there's a reason that there's an iris diaphragm. But if you want more light to come in, you would just open the iris diaphragm. So we need to talk about a few things that happens when using our microscope. And so we're going to answer some questions together. So let me put my pen on. So the question says, as magnification increases, what happens to the working distance, which is the distance between the objective lens and the slide? So if this is my 4x, and here's my slide, that's my slide. And now I go to my 10x, and there's my slide, right? And so my 10x objective is going to be a larger lens. So what happens to the working distance, the distance between the slide and the lens, as we go up in magnification? And that is that you get a decrease in the working distance. Because larger lens. Right? So notice that your slide is now much closer to that lens. Now, the reason that I bring that up is because the slide and the lens get closer, this is why you should only use fine focus when increasing magnification. So you should only use the fine focus when you increase your magnification, because you'll notice that as your lens gets larger and larger, the distance between the lens and the slide starts to get really close. And if you accidentally use the 40x, or I'm sorry, if you accidentally use the coarse focus, and you move the slide and the stage more rapidly, you risk possibly snapping the slide, or even worse, snapping the lens if the two get too close, especially if you're using an oil immersion lens. So because our working distance gets smaller, because that distance gets smaller, we only want to use the fine focus when we go up in magnification. So the next question says, as magnification increases, what happens to the light intensity? And so you have to remember that the condenser is going to pass light through the specimen. Now, if you think about this and you know about what happens to light when it hits glass, so if the light hits the glass, what is going to happen? And the answer is that when light hits glass, it's going to refract. So it's going to go all over. Notice when we go up in magnification and our lens gets larger, less of that light is getting up and into the lens. So as our magnification increases, what happens to light intensity? We get a decrease in light intensity. Need to adjust to allow more light through. So again, because that lens gets closer to the slide, less light is getting up and into the lens. And so as we go up in magnification, we are going to decrease our light intensity. And then the last question on here asks, as magnification increases, what happens to the size of the field of view? And so what that refers to is if I'm looking at my slide, okay, and I'm looking at my field of view, when I look at my letter E, and let's say that this is at 40x total magnification, when I go to 100x, my letter E is going to take up a much greater view. And when I go to 400x, I might only now see a portion of that letter E, meaning I might only see that line that comes across, but I don't see the whole letter E. So as my magnification increases, right, as I'm zooming in, what happens to the field of view? It decreases field of view. Because we're increasing magnification, we're zooming in, now we're not seeing as great of an area. This is why we start with 4x objective. So we start with the 4x objective, meaning a total magnification of 40x, because we have the greatest field of view, meaning it makes it much easier to find our specimen. If we tried to start at 400x total magnification and we're so zoomed in, it's going to take a while to find where that specimen actually is. So as our magnification increases, our field of view decreases. So notice that magnification is inversely related to all of these other things, meaning as magnification increases, working distance decreases, light intensity decreases, field of view decreases. They're all inversely related. So when you're doing this, what we would do in lab is we would prepare several slides to look at under the microscope. And so one of the specimens that we would do would be the Elodea. And the Elodea is a plant. You might recall using it in your photosynthesis lab. It's a freshwater plant, and it looks like this diagram here on the right. And so what you would do is you would take a glass slide, you would add a drop of water, you would take a small portion of the leaf, you would put it in the water, and then cover with a cover slip. You would view it under low power and then go to high power. So what you would do is you would look at it under a total magnification of 400x. And what you would do is you're going to draw for this in your lab, you're going to draw one cell in pencil, so do this in pencil, large enough to see the detail. And you want to make sure to label your drawings and make sure to indicate the magnification. So when you do this, you would want to indicate your magnification should be 400x, and you need to label several parts. So we have our chloroplast, we have our cell wall and our cell membrane, and we have our cytoplasm. So if I look at this picture on the bottom, what do you think, what part would be the chloroplast? And so if you said these green dots, that would be right. The chloroplast is an organelle that plants use to do photosynthesis. And so we saw that in our photosynthesis lab. Next, it says to label the cell wall and cell membrane. In a plant cell, the cell wall and cell membrane are right next to each other. And so that border there is your cell wall / membrane. They're right next to each other, and we can't distinguish between them. And then lastly, we would label the cytoplasm. So the cytoplasm is the inside of the cell, but outside of the chloroplast. So this would be our cytoplasm. And so when you do this, though, don't draw all these cells. Draw just one cell, label the parts. So just like I've done here, but draw one of those cells only. The next part, and this also goes with a question in the lab, and that is that we're looking at Elodea that's undergoing what's called plasmolysis. So what we would do is that same slide that we had before, we would take the cover slip off, and we would add a drop of salt water to the slide. Now, we have a lab later on looking at osmosis. But one of the things that we're going to learn is that if we look, if this is our plant cell, and we add salt water. So what's going to happen is we have a lot more salt outside. So the purples are salt. We have a lot more salt outside relative to the inside. And so what that means is that we have more free water inside. So inside has more free water. And so what's going to happen as a result is because the concentration of water is higher inside, the water is going to move out of the plant cell. So the water is going to come out. And what ends up happening is as a result, the cell membrane starts to shrivel and shrink in, because you have to think about the fact, so if you look here, you have to think about the fact that for a plant cell, they have a cell wall and a cell membrane. Cell wall is a very rigid structure. The cell membrane is very fluid. So as the water comes out of the cell, what's going to happen is that the cell membrane is going to collapse in. And so your lab has a question about what structure can you see as a result of plasmolysis. And the answer is you can see the cell membrane, because now the cell membrane separates from the cell wall. So now if we go back and we label this, so we have chloroplast. So all of those little green dots in there would be our chloroplast. We have our cell wall. Out here, see where all these chloroplasts have clumped together? That now is all the cell membrane. So that red outline that I drew, that's now the cell membrane. We can now see the cell membrane because by placing the plant cell in salt, that's going to cause the water to come out of the cell membrane is going to collapse in, and now we can visualize where the cell membrane is. And then the cytoplasm would be the space inside the cell membrane, but outside of where the chloroplasts are. So the cytoplasm would be in here, this area here, not the cytoplasm. Okay, and so you're not going to label that the cytoplasm. The cytoplasm is inside here. So when you do this, you want to draw just one cell undergoing plasmolysis and label the following parts. And again, your total magnification for this, 400x. The next slide that you would look at would be your onion skin. Onion cells have an epidermis, just like our skin has an epidermis. And so what that is if you've ever peeled an onion, you know how onions have like rings. And so the skin is going to be the inner side of one of those rings. And so what you would do is you would take a glass slide, you would place on it an onion skin. So a thin piece of the onion. You would add 2 drops of methylene blue, let it sit for 5 minutes, 5 drops of alcohol, let it dry, 4 drops of xylene, and then add a cover slip. Now, the alcohol and the xylene help with dehydrating the slide and making it easier to see. So you put the cover slip, and you either would view this using your 10x objective or your 40. So that's why it says view under low power or high power. So either would be fine, okay? Depends on how it looks on your microscope. But what you're going to see is here is your onion cell. So where do you think the nucleus would be? And the answer is this dot is the nucleus. The nucleolus would be a darker dot. So there's a dot usually within there. That's the nucleolus. And the nucleolus is the site of ribosomal RNA synthesis, as well as where ribosomes are assembled. We have our nuclear membrane. So that's the outer edge of the nucleus. We would have our cell wall / membrane. We can't distinguish it at this point. And we would have our cytoplasm. So notice that when you look at this plant cell, when you look at the onion skin, do you see chloroplasts in here? And the answer is no. Notice you don't see those green organelles in the onion cell. That's because not all parts of the plant have chloroplasts to do photosynthesis. The leaves are where photosynthesis is going to take place. The onion, which is the root, is not going to have photosynthesis take place, right? It's underground, it's not seeing sunlight, therefore it's not doing photosynthesis. And so that's why you see this plant cell that does not have a chloroplast. And then lastly, we have our animal cheek cell. And so what you would do for this is you would take a glass slide, add 1 drop of methylene blue, and then you would take a sterile toothpick and you would open your mouth and say, ah, and you would gently take the toothpick and scrape the inside of your cheek. Now, we don't need blood cells, so don't stab yourself with it. However, if you just take your toothpick and lightly touch the inside part of your cheek, so in your mouth, and you just scrape the toothpick along the inside of your mouth, you're going to pick up some of these cheek cells. You take that toothpick, you swirl it in the methylene blue. You would dispose of the toothpick in the trash, so we would not want to leave those out. You would then take your cover slip your slide, view under low power and then high power, and then label the drawings. And so it says to label your drawings the nucleus. So that's that dot here. We would have our cytoplasm. And then we would have our plasma or cell membrane, right, which is this outer edge of the cell here. So you need to draw one animal cheek cell, label nucleus, label the cytoplasm, and label the cell membrane. And so your total magnification for this would again be 400x. And so those would be the slides that you would have prepared in lab and viewed under the microscope. And for the purpose of this, since obviously you're not in class, you can just draw from the images that are provided. So the next part of the lab is looking at mitosis. And mitosis is the process by which the cell goes through what we call the cell cycle. And its job is to produce two exact copies. And so what that means is that you start with one cell, and we're going to call this cell the mother cell. And the mother cell divides and gives rise to two identical daughter cells. Those two offspring are exact copies of the mother cell. Now, the purpose of mitosis is that it's essential for several reasons. One is mitosis is used for reproduction, and this is used for single-celled organisms. And we call this type of reproduction asexual. Asexual refers to without a partner. So an organism like you can see on the bottom left, an organism like an amoeba would reproduce by mitosis. They reproduce asexually by simply just making a copy of themselves. Next, mitosis is also important for growth and development. And this is true for multicellular organisms. So if you think about yourself for a minute, you started as one single cell. You had an egg fertilized by sperm. You end up with a single-celled zygote. That one cell then divides into two. Those two cells divide to 4, 4 divide to 8, 8 to 16, and so on and so forth. And so the way that you go from being a single-celled organism to a multi-celled adult is through mitosis. So along those lines, if you think about it, think of your own body, right? Within your body, you have a variety of tissues. You have muscle cells, you have liver cells, you have blood cells, etc. And so those different cell types are different in your body. But what I want you to think about is, what is it that makes them different? And so I want you to think, because conceptually students struggle with this a little bit or have misconceptions about this, and that is, if you think about your cells, all the different cell types in your body, do those cells have the same DNA, or do they have different DNA sequences? And so think for a minute, all the cells in your body, minus your reproductive cells, all the cells in your body, do they have the same DNA? And if you said yes, that's correct. So your skin cells have the same DNA sequences as your liver cells, and your blood cells have the same DNA sequences as those as well. And that's because all the cells in your body arose through mitosis from one single cell. What makes those cells different from one another is what we call differential gene expression, meaning each cell has the exact same DNA, but what makes them different is what genes or what DNA sequences are turned on or off, meaning what proteins are being made in the cell. So if you look at a skin cell would be turning on expression of keratin. Keratin is a tough structural protein that's used in skin. That same gene is in blood cells, but it's not being expressed. Red blood cells, on the other hand, would express or turn on expression for hemoglobin. Hemoglobin is a protein in red blood cells that is responsible for carrying oxygen to all the tissues in the body. And so I just wanted to point out that you want to think of all the cells in your body have exactly the same DNA sequence. And that's because those cells arose, you became a multi-celled organism through mitosis. Each cell went through and divided and gave rise to two identical daughter cells. The last reason that mitosis is used for is for tissue renewal. So that is replacement and repair of damaged cells. So think about if you were to get a cut, for example, right? Let's say my arm, I got a cut. Well, if that skin is damaged, does that cut stay open forever? And the answer is no. And that's because the tissue gets repaired. And it gets repaired because the cells in the area will undergo mitosis and they will make an exact copy, and that will repair the tissue. So if we look at the cell cycle can be divided into two main phases. The first phase is interphase. Interphase is where we're going to have the most metabolism and growth. And so notice that about 90% of the time, the cell is in interphase. So if we look at interphase, interphase is divided into three subcategories. The first one is called G1. G1 stands for gap 1. And during gap 1, or G1, this is where we get the synthesis of macromolecules and cytoplasmic organelles. So this is where the cell is synthesizing its proteins, it's making lipids, etc. So it's making its macromolecules and it's starting to replicate its organelles. Now, S phase is for synthesis. This is where DNA gets synthesized or replicated. So what you're looking at is this is a chromosome. So that's a chromosome. And during S phase, that chromosome is going to duplicate and it's going to give rise to what are called sister chromatids. And sister chromatids are exact copies of one another. So if I look at the one on the left, it has the exact same DNA sequence as the one on the right. And that's because those sister chromatids are going to be exact copies, because that in S phase is where DNA gets duplicated. So it's going to make an exact copy. After S phase, we have G2. G2 is just a phase for additional growth, and the cell is getting ready to divide. After interphase, we have what we call the mitotic phase. This is the cell division part of the cell cycle. And so this is spent about 10% of the time. So this happens much faster. So the mitotic phase is a much smaller percentage of time. And mitosis is where we're going to divide the nucleus, meaning we're going to separate sister chromatids. Mitosis can be divided into 5 subphases. Sometimes you'll hear it referred to as 4. And that is PMAT. The way I remember that is PMAT, prophase, metaphase, anaphase, telophase. Some groups have prometaphase, which is why it says 5 subphases. So mitosis is going to divide the nucleus. After we divide the sister chromatids, we're going to undergo cytokinesis. Cytokinesis is going to divide the cytoplasm. This is where we physically divide. So notice here, these cells are still connected, but cytokinesis is going to physically divide those cells into 2. So we started with one daughter cell and we ended with 2 identical daughter cells. So we started with one mother cell. Now, in order to understand what happens in mitosis, we need to talk a little bit about chromosomes. So again, if we look here, this is our chromosome. And so this would be our chromosome in, let's say, G1. G1 is gap 1. Now, again, during S phase, in S phase, DNA is going to duplicate. So this would be like a cell in G2, so after S phase. And what you end up with is you have two sister chromatids, and they are exact copies of the parental. So these two DNA sequences are identical. They are held together at a portion that's referred to as the centromere. The centromere is basically a DNA sequence where proteins are going to hold those sister chromatids together. So after S phase, each chromosome is made of two sister chromatids. Now, one of the things you're going to see in a minute is that when the DNA is in the nucleus, and it's in the nucleus during interphase. In interphase, the chromosomes are not tightly compacted, meaning they're very loosely associated in the nucleus. The reason for that is that when the DNA looks like this black and white electron micrograph, when the DNA is in that configuration, the DNA is packed in tightly. And remember that I said in interphase, this is where a lot of metabolism occurs, meaning a lot of gene expression occurs. When the DNA is packed in tightly, like you're seeing in this other picture, that type of DNA structure does not allow gene expression to occur. And so in interphase, the chromosomes are very loosely compacted. They're not packed in real tightly because the DNA is unwound and it's being transcribed and translated, which you guys will learn about later. Now, if you think about it though, in interphase, if the DNA is very loose, think of it like a ball of yarn, right? So if I have my ball of yarn and it's not very organized, it's all just kind of loosely assembled. If I go to try and separate that ball of yarn, it's not going to be an easy task to accomplish because there's no organization. However, what happens is before those sister chromatids can separate, so before the DNA can move to opposite sides of the cell, the chromosomes have to condense. They have to pack in tightly. They need to pack in tightly so that they can prevent tangling, meaning that when we go to split those sister chromatids, we need to have it organized. So this would be analogous to, again, back to our ball of yarn. If we organize it and we wind it around a spool, it's going to make it much easier to separate than if we just have this big tangled mess. And so one of the things that's going to happen is once the cell begins to do mitosis, chromosomes are going to compact, they're going to condense, they're going to pack in tightly. And then when we go through mitosis, now our sister chromatids are going to separate. And so what you're left with is you have two daughter cells, and these daughter cells have the sister chromatids separated, and they are exact copies of this parental cell. So now we're going to walk through and we're going to talk about the steps of mitosis. In addition, on Canvas, I'm also posting a video that summarizes this so that you can see it in an animation as well. When you're going through this part of the lecture, you want to turn to page 91 of your lab and use this explanation to help you write what happens in each step for question number 4. So question number 4 basically asks you to describe what happens in each of these steps in mitosis. So again, if we're talking about the mitotic cell cycle, the first thing that's going to happen is going to be interphase. interphase, remember, is divided into 3 phases: G1, S, G2. And so again, what's going to happen in interphase is macromolecules are going to be synthesized. So that's one thing you want to include. We have synthesis of macromolecules. And then we have chromosome duplication, DNA synthesis, right? Because in S phase, that is where DNA is going to replicate. We also have this structure inside the cell that's called a centrosome. And the centrosome is this T-shaped structure. It's made up of centrioles. These are structures made of microtubules. And they're going to act as what we call the microtubule organizing center. And what that means is that during the cell cycle, we're going to have these spindles that are going to come out from the centrosomes. Those spindles are also made of microtubules, and their job is to help move chromosomes within the cell. So you'll see that in a minute, but during interphase, we get duplication of centrosomes, because what's going to happen, you're going to see during mitosis, these centrosomes are going to go to opposite ends of the cell. So in interphase, we have our centrosomes that are being replicated. So now, when we are looking at our phases of mitosis, again, depends on who you ask. Some people will divide mitosis into 4 phases, and again, those 4 phases would be PMAT. It's the easiest way I can remember that. Prophase, metaphase, anaphase, and telophase. You'll notice that sometimes people will separate out prometaphase as its own phase. For the purpose of lab today, all the events that you see in prometaphase, go ahead and lump it into prophase. So we're going to condense that and just call that all part of prophase. So some people will call it early prophase and late prophase. Some will actually call it a separate prometaphase. But for the purpose of this, we're going to put prophase and prometaphase in the same category. So what happens during prophase? So remember that I said in interphase, the DNA is loosely compacted, right? It's not packed in tightly. So in prophase, this is where chromosomes condense. They're going to start to pack in tightly, and they're going to make that characteristic X-looking shape that we're used to seeing. Another important thing that happens in prophase is that the mitotic spindle begins to form. And so again, that mitotic spindle is made of microtubules, made of this cytoskeletal element, and those microtubules are going to play a role in moving chromosomes within the cell. So we have our centrosomes, now we have our mitotic spindle beginning to form, so we have our microtubules coming out. In prometaphase, we're going to get the nuclear envelope breaking down. So we're going to break down that membrane for the nucleus. The spindle fibers are going to make contact with the chromosomes at a structure that's called the kinetochore. And the kinetochore is basically a protein structure that the spindle is going to attach to. And when the spindle attaches at the kinetochore, that allows the cell to be able to move the chromosomes within the cell. So prophase, and including prometaphase, the cell is preparing for division. Chromosomes condense, mitotic spindle forms, nuclear envelope breaks down, spindles attach to the chromosomes. So those are the big events that happen in prophase. Metaphase, okay, in metaphase, think middle. So what's going to happen in metaphase is that the chromosomes are going to line up along the metaphase plate. So they're going to line up along the metaphase plate, which is this imaginary equator in the cell. It's the middle of the cell. And you'll notice that the way that they line up is each sister chromatid faces a different centrosome. So notice that again, we have two centrosomes. They're at opposite ends of the cell. They moved towards opposite ends of the cell in prophase. In metaphase, when they're at the opposite ends of the cell, that's when the sister chromatids are going to line up single file along the metaphase plate so that a sister chromatid faces each of the centrosomes. Next, we have anaphase. In anaphase, this is where sister chromatids are going to separate. So they're going to get pulled to opposite ends of the cell, and the cell is going to elongate. So the cell elongates with non-kinetochore microtubules. So you'll notice that some of these microtubules, that one's not a very good arrow, some of these microtubules are not attached to a chromosome. And the ones that are not attached to a chromosome, we call those non-kinetochore microtubules. And so those microtubules are the ones that are responsible, they push off of each other, and they're responsible for the elongation of the cell. Basically, they're going to elongate the cell so that the cell can then eventually divide into two daughter cells that are copies of the parent. So the big thing though in anaphase, sister chromatids are going to separate. The sister chromatids are going to move towards opposite poles of the cell. Then we have telophase. And the way I remember what happens in telophase is that telophase is the opposite of prophase. So basically everything that you said for prophase, the opposite happens in telophase. So let's walk through that. So prophase, chromosomes condense, which means in telophase, chromosomes decondense. They decondense or they unwind. Right? So again, prophase, we said chromosomes condense. Telophase, they decondense. In prophase, nuclear envelope breaks down. Telophase, nuclear envelope reforms. Prophase, spindle, the spindle apparatus forms. Telophase, spindle apparatus disintegrates, right? It's going to break down. And so again, telophase you can just think of as the opposite. Telophase is the opposite of prophase. So everything that happens in prophase, the opposite is going to happen in telophase. Now, while the cell is undergoing telophase, simultaneously while it's doing telophase, you're going to see it undergoing cytokinesis. And again, cytokinesis is going to allow the cell to divide the cytoplasm. And so the cytokinesis is going to occur when the cytoplasm is split. And so you're going to see in a minute the differences between how plant cells do cytokinesis and how animal cells do cytokinesis. So when you're taking notes about what happens during cytokinesis, it's not enough to just say it's division of the cytoplasm. I want in your answer a little more detail about how that happens in animal cells and how that happens in plant cells. Now, what you have to think about is to think about the structure of those cells. Animal cells, do they have a cell wall? The answer is no. They have a cell membrane, but they don't have a cell wall. Plant cells do have a cell wall, and their cell wall is made of cellulose. So the way that plant cells and animal cells divide their cytoplasm is different because plant cells have a cell wall, animal cells do not. So what you're going to see is that if we look at the differences between plant and animal cells are going to divide using a cleavage furrow. And so what that means is that you're going to notice we have this contractile ring of microfilaments, which is also called actin. It's a cytoskeletal element. And so those actin filaments are going to form a ring around the middle of the two cells. And so you can see this in this electron micrograph of an animal cell. You can see this cleavage furrow. And so these actin filaments are going to form this ring around the cell. And one of the things that you'll learn is that cytoskeletal elements, actin for example, works with a motor protein. And in this case, the motor protein is called myosin. Actin and myosin also work together for muscle contraction. And that's because myosin, the motor protein, is going to walk along the actin and it causes it to contract. Same idea here for the cleavage furrow. Myosin and actin are going to work together. They're going to contract and they're going to physically divide that one cell into two. Think of it like it's pinching it off. It's going to pinch down the middle and it's going to pinch it until you end up with two daughter cells. So that's for animal cells. Plant cells, on the other hand, have to do it a little bit different because they have a rigid cell wall. So for them, a cleavage furrow is not going to work because the cleavage furrow has to pinch the cell membrane. Cell membrane is a very fluid structure. That's easy to do. Not so much for a cell wall, which is very rigid. So plants do it a little bit different, right? And so for a plant cell, for example, a plant cell is going to form a cell plate instead. And so you can see down here, here's this picture. Here is in the middle, there are these vesicles forming. So these are these little transport vesicles, and in them they have the components for a new cell wall and cell membrane. Those vesicles are going to fuse until you get what's called a cell plate. It's this line that's forming down the middle, and that line is going to have the cell wall and the cell membrane. And then that cell plate is going to continue to extend until it reaches the cell wall. And what you end up with is you end up with a new cell wall down the middle that divides that one cell into two. And so there's a question in your lab that asks you about differences between plant and animal cells, right? It asks you to list some of the differences between plant and animal cells. Here's one, right? The way that they do cytokinesis is different. Animal cells use a cleavage furrow. Plant cells use a cell plate. There's one difference. There are two other big differences that we've talked about. Look back at our slides about the Elodia, for example, versus the human cheek cell. And there are some structures that you'll notice that plant cells have that animal cells do not. And if you think about it, it is along the same lines as to why they do cytokinesis different. Plant cells have a cell wall. Animal cells do not. So there's another difference. You also might recall that plants are what we call autotrophs. They are self-feeders. They're able to make their own food because they have a organelle that animal cells do not, and it's that little green organelle. Do you recall what that little green organelle is called? And hopefully you remembered that that is the chloroplast. So plant cells have a chloroplast. Plant cells have a cell wall made of cellulose. Plant cells divide their cytoplasm they divide their cell wall by using what's called a cell plate. So they do cytokinesis using a cell plate. So what you're going to do is there's a place in your lab, so on pages 89 and 90, there's a place for you to draw an example of one cell that's in each of the phases. So you need to draw one cell that's in interphase. You need to draw one cell that is in prophase, one cell that's in metaphase, one cell that's in anaphase, one cell that's in telophase. So only draw one cell per box. And let's kind of walk through this. So if we look at interphase, here is a cell in interphase. The purple is going to be the DNA. And so notice that you don't see any distinguishable chromosomes at this point. The chromosomes are decondensed, right? They're not packed in tightly. So you can see that this cell is an interphase. This little line here, that's the cell wall and the cell membrane. Remember that you cannot distinguish them at this point. The purple circle here is the nucleus, which means that the cytoplasm is the space outside of this light purple, but inside the cell wall. This little circle here, this is going to be our nucleus. The darker purple dot is going to be the nucleolus, and that would be our cell in interphase. Now, if we're trying to locate a cell that is in prophase, remember that what's going to happen in prophase, the main events that happen in prophase, chromosomes condense, so you can see that they're starting to pack in more tightly. Nuclear envelope is going to break down. So notice that in this cell, right, the DNA is still kind of in a circular shape because the chromosomes are condensed and the nuclear envelope is starting to be broken down. So this would be an example of prophase. So we don't really have a nucleus anymore, but we do have chromosomes. The edge here is your cell wall and cell membrane, and the space in here is going to be your cytoplasm. So that's your cell that's in prophase. Metaphase, when you look at a cell that's in metaphase, remember that metaphase means middle, right? So metaphase middle, that is where the chromosomes line up single file along the metaphase plate. Now, one of the things that you want to think about is that while textbooks like to show it as this perfectly straight line, in actuality, that is not what it really looks like. And that's because the middle part of the chromosome, remember that the chromosomes are held together at the centromere, so only part of the chromosome is held together. The other parts, the other arms, are free floating. So think of it like your own body. And think of it like your torso is going to be your centromere. That's the part that's holding the two halves together. Your left side of your body, your left arm and your left leg, is one sister chromatid. Your right arm and your right leg is your other sister chromatid. Notice my middle is stuck together, but my arms and legs don't have to be stuck together. They can go off to the side. That's what you're seeing here. So you're seeing that the centromere is lined up in the middle, but the arms and the legs can go off to the side. So what you're looking at in this picture, the purple again, those are the chromosomes. We have our cell wall and cell membrane. You can't distinguish between them. And then we have the cytoplasm, which is the space in here. So if you look at this other in this diagram, can you locate one other cell that is also in metaphase? So look around and see if you can find another example of this. And one of the best examples I can see is this one here. It's not quite in focus, but you can definitely see that they're lined up in the middle. So metaphase is the middle. They're lined up along the middle. Anaphase is where sister chromatids are being pulled to opposite ends. So this would be an example of a cell that's in anaphase. So what you have is you have your cell wall, that's the edge. We have the purple being the chromosomes, and you can kind of see very faintly those like orangish color lines that are coming out. Those orangish color lines are going to be your spindle. Those are your spindle fibers that are responsible for pulling chromosomes to opposite ends of the cell. Now, again, the spindle is made of microtubules. So one thing to think about is that some of our chemotherapy drugs, some of our cancer drugs, target microtubules. And the reason for that is that cancer is uncontrolled cell growth. What has to happen during cell division is that chromosomes have to move to opposite ends of the cell. What has to happen for that to occur is that those microtubules have to elongate and they have to shorten. That's what's going to help to pull the chromosomes apart and pull them to the opposite ends of the cell. So microtubules are what we call very dynamic. They're constantly changing. They're growing, they're shortening, they're growing, and they're shortening. And so drugs that target microtubules, like if you've ever heard of a drug called Taxol, the way that it works is that it inhibits microtubule dynamics. So it prevents microtubules from being very dynamic, which then prevents chromosomes from being able to move, which then prevents cell division. Now, the problem with that approach for chemotherapy is, does that only target cancer cells? And the answer is no. It targets all rapidly dividing cells. And so this is where a lot of side effects come for in chemotherapy, right? If you think of common side effects that you see with chemotherapy drugs, hair loss, because your hair, those cells are cycling. So when you inhibit microtubules, the patient is going to lose hair. GI distress, so gastrointestinal stress, so your stomach, the lining of your stomach gets very irritated from the cells not being able to divide, etc. And so that's just to get you to think along these lines of practical applications of this, is that when you're talking about cancer therapies, right, you're talking about inhibiting cell division. And so one way that we can do that is by targeting microtubules, because microtubules play a role in the spindle, which is going to play a role in cell division. So that's anaphase. Here's another example of anaphase up here. So there's another example of anaphase. Next, we have telophase. Again, telophase is the opposite of prophase. What you can look for telophase is, notice this little line, this orange line that's here. That orange line is going to be your cell plate. That's the cell plate that's starting to form that is going to divide that cell into two. And so again, you might recall that one of the things that's going to happen is that the chromosomes, when they're at opposite ends, chromosomes are going to decondense. Then the nuclear envelope is going to reform. So you're starting to see less distinct chromosomes. They're starting to become more circular. And while the cell is going through telophase, it is simultaneously also going through cytokinesis. So they're occurring at the same time. So we're seeing the cell plate form at the same time that we're seeing telophase occur. Again, our cell wall, cell membrane is the outside of this little box. Again, the cell plate is that line. These orange lines coming from the chromosomes and into the cell plate, those are the spindle fibers. So that's where you would label the spindle fibers, etc. And so when you do this, you're going to take this picture, and for each phase, you're going to draw one example. So for interphase, you're only going to draw this one cell. Do not draw this whole thing. Draw this one cell and then label the parts on that page that has everything you need to label. So make sure that you cross out as you label it so that you don't miss any labels. So you're going to draw and label one cell in interphase. You're going to draw and label one cell in prophase, one cell in metaphase, one in anaphase, one in telophase. Here's another example of a cell that's in telophase. Again, you can see the cell plate forming. Now, there is another question in your lab on page 92. And what that question is looking at is, for example, if you were to count 1,000 cells. So this is an onion root tip, and the onion root tip is used because the root of the plant is going to be dividing. So this has the cells that are going to be in the mitotic phase. If you look at these cells, notice how many of them are in interphase, right? Interphase, 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 interphase. And again, that makes sense because 90% of the time, the cell is in interphase. And so again, one of the ways that this can be done is if you look at a sample like this, you could quantitate or count the number of cells that are in each phase. And you could keep a tally. And you would see, let's say 90% of those cells would be in interphase. So if you were counting 1,000 cells, 900 of them would be in interphase. And so what that question on page 92 is asking you is, based on that data where they counted the number of cells in each phase, based on that data, what is the longest stage of the cell cycle? The cell cycle includes interphase and the mitotic phase. And so of those prophase, metaphase, anaphase, telophase, and interphase, based on the percentage of cells that are in a particular phase, what is the longest phase of the cell cycle? Well, that's going to be the one that has the most number of cells, because if you see the highest percentage of cells in a particular phase, what that tells you is that the cell spends most of its time in that phase. So when you're answering that, make sure to read carefully between if it's asking for the cell cycle, which includes interphase, or is it asking for the phase in mitosis, which is going to be different. So keep that in mind when you are answering that question. So that's what you're going to do for your onion root tip. Draw one of each cell and label the appropriate parts. The next part is going to be looking at a white fish blastodisc. And so in your lab, these are actually drawn for you. They put an example of one that's in prophase. They put an example of one that's in metaphase, so on and so forth. And so for that one, you don't have to draw what those look like, but they're already there for you. You just need to label the parts. So again, use that page, that one page that has a summary that tells you what to label. And so you can also use this to kind of practice to see if you can identify these different stages. And so this is just showing you a different picture of that. Now, if we were actually doing the lab, it's important that you understand how to put away the microscope properly. So I left this in here because if you go to use a microscope again, it's important that you know the proper steps to clean up the microscope. And so one of the first things you always want to do is you always want to make sure to put the 4x objective back in place. That's the smallest lens, right? And the reason is we don't want to put the biggest lens back and risk that the stage could hit it. So we always put the 4x objective in place. We would raise the stage to the top. Now there is some debate. Some people raise it, some people lower it. For the purpose of this, we would raise it to the top. We would center those stage clips. So those metal parts that hold the slide, we would want to center those so that they're not hanging over one end. Because if they're off to one side and you happen to hit them, you might break it. So we want to make sure that the stage clips are in the middle and they're not hanging over one side or the other. We would want to turn down the rheostat, so turn the light intensity all the way down. and then turn off the switch. Again, because remember we talked about that when we go to turn on our microscope, before we even plug it in, we want to make sure that the switch is off and the light intensity is down. So same idea when you put it away. You want to make sure that the light intensity is all the way down, then turn off the switch, and then unplug it. And then that's ready for the next person to use it. You would wrap the cord around the back part of the microscope. You would put the cover on it. Don't worry about how you would dispose of the slides, but I just wanted to make sure to still cover how to clean up your microscope. And then this is your Quiz 6 study guide. So you guys can read through this and make sure that you know these different parts in order to study for Quiz 6. And so now hopefully this video should allow you to be able to do Lab 6 on your own without being in lab. And make sure for Quiz 6 that you also read Lab 7 ahead of time and you read about meiosis and human genetics. And so this concludes this video.